Hello friends and welcome to my channel Sequential Biology. In this video we will discuss about process of spermatogenesis. The spermatogenesis is the process by which spermatozoa are synthesized or produced inside male gamete or testis. Remember, gonad is a reproductive organ and the gamete is the product of this reproductive organ. And you know the sperm is a tadpole-like cell and this is haploid cell. And it has four specific regions: the head, neck, middle piece and the tail region. And in head portion the nucleus is present and the chromosome in nucleus are haploid. Just in front of this nucleus the acrosome is present and this is made up of different type of enzymes. And next part is middle piece, and in middle piece the spiral mitochondria or Nebenkern is present. After middle piece the tail region is present, and in this tail region this is flagella-like structure, show our spermatozoa are motile structure. Now, these spermatozoa or the sperm are synthesized in human testis, but these are collected in epididymis. And if we want to collect the spermatozoa, then we should search the epididymis, not the testis, because the sperm are stored inside epididymis. Now, what will be the different stages of spermatogenesis? And you know the gamete are synthesized by process of meiosis, and the meiosis again occurred by two phases: the meiosis 1, which is known as reductional division, and the meiosis 2, which is known as equational division. But the primordial germ cell is present during our embryonic stage, and this primordial germ cells undergo mitotic division, and this will produce many spermatogonial cells. This spermatogonial cell is a kind of stem cell, that means it have dual properties, division and the differentiation. And since it is stem cell, so the stalk of the spermatogonial cell, stem cell, never ends. This spermatogonium, by process of mitotic cell division, this will produce the primary spermatocyte, and this primary spermatocyte undergo the meiosis 1. And here you can see the primary spermatocyte is diploid, and the meiosis 1 is reductional division, so by process of meiosis 1, the secondary spermatocyte are produced, and this secondary spermatocyte is haploid because this meiosis 1 is reductional division. And then the secondary spermatocyte undergoes meiosis 2, and this will produce the early spermatid. And here you can see these spermatid are a rounded cell, but this will be changed into a tadpole-like structure. So huge changes occurs in this spermatid, and which causes the development of tadpole-like spermatozoa. And we will discuss about these different stages. The germinal epithelium produces the primordial germ cell, and this primordial germ cell differentiation within the testis, and this will produce the spermatogonium cell. And from this spermatogonium cell the spermatozoa are developed, and this spermatogonial cell rests on the basal membrane of the seminiferous tubule. And we know inside the testis the two important structures are present: the seminiferous tubule, these are the seminiferous tubule, these are the tubule-like structure, seminiferous tubule, and in between the seminiferous tubule a special kind of cell are present, and which is known as Leydig cell. And the spermatogenesis actually occurs in this seminiferous tubule. Then from the germinal epithelium the primordial germ cells are developed, and this primordial germ cell differentiated within testis, and this will causes the development of the spermatogonium cell, and this spermatogonium cell rests on the basal membrane of the seminiferous tubule. And from this spermatogonial cell, by different stages, the mature spermatozoa are developed. Development of spermatogonium to the sperm is known as spermatogenesis. So development of spermatogonium to sperm, this stage, development of spermatogonium to sperm, is known as spermatogenesis. And this spermatogenesis occurs in two phases: the formation of the spermatid from the sperm mother cell or the spermatogonium. Formation of the spermatid from the spermatogonium, this is the first step, and the second step is formation of spermatozoa from the spermatid. And you know the spermatid is a round cell. A huge changes occurs in this spermatid, and this will causes the development of a tadpole-like spermatozoa. Here are the different stages of spermatogenesis process. The primordial germ cells undergo the repeated mitotic division, and this repeated mitotic division causes the formation of spermatogonium cell. and there are present a different spermatogonium cell, but the last spermatogonial cell nature will be changed, and instead of division it grows. Then the spermatogonial cell undergoes the growth, and here you can see the spermatogonium cell, the G. So G means growth. That means the cell which grows to form the primary spermatocyte, this is known as spermatogonium cell. Then the growth of the spermatogonium cell causes development of primary spermatocyte, and this primary spermatocyte undergo meiosis 1. This is primary, so meiosis 1, and this causes the development of secondary spermatocyte, and this secondary spermatocyte is always haploid, and this primary spermatocyte is diploid. Then the meiosis 1 is also known as reductional division, and this haploid, the first form haploid cell is secondary spermatocyte, and this secondary spermatocyte also undergoes meiosis 2 division, and this meiosis 2 division is equational division, and each cell produce 2 cell, right? And this will causes the formation of spermatid, and spermatid is a rounded cell, and then a huge changes occurs in this spermatid, and this spermatid will be changed into tadpole like spermatozoa, and the process by which the spermatid are changed into the mature sperm is known as spermiogenesis. Then the spermatogenesis occurs in 2 phases: the formation of spermatid from the spermatogonium cell, this is the first stage, and the formation of mature sperm from the spermatid, this is the second stage. And these 2 stages occurs successively, and this will causes the formation of million sperm in our gonad or the testis. Then the spermatid, by process of spermiogenesis, its shape will be changed, and here you can see the spermatid is a round structure, and this round structured spermatid will be transformed into tadpole like spermatozoa. Then the spermatid will be elongated and sheds excess cytoplasm and develop the tail. These are the important events which occurred during the time of spermiogenesis. Then the spermiogenesis is the process by which a spermatid changed into the spermatozoa. Now we will see the process of spermiogenesis. The spermiogenesis occurs in different phases. The first phase is Golgi phase, and in Golgi phase contains, and this cell, and in this phase the spermatid contains Golgi complex, numerous mitochondria, smooth endoplasmic reticulum, and pair of centriole. So this is a spermatid and which remains in Golgi phase, and in this phase you can see the Golgi complex, mitochondria, pre-acrosomal granules, and the centriole. These are present in Golgi phase, and the next phase of spermiogenesis is known as acrosome phase, and the pre-acrosomal granule, this pre-acrosomal granule fused to form the acrosome vesicle over the nucleus. We know the acrosome cap are present just over the nucleus, and this acrosome vesicle are formed due to union of this pre-acrosomal granule, and the acrosome vesicle contains many hydrolytic enzymes like hyaluronidase, neuraminidase, trypsin like protease, acid phosphatase. These are the different enzymes which are present in acrosomal vesicle, and also the centriole form the axoneme of the flagella. So this centriole present during the Golgi phase, and in acrosomal phase, from this centriole the axoneme of the flagella are developed, and the mitochondria around the proximal part of flagella. In proximal part, that means the anterior part is surrounded by mitochondria, and later this will form the spiral mitochondria. And the last stage of the spermatogenesis is maturation phase, and the remaining part of the Golgi, so these are the remaining part, these are excess cytoplasm, this excess cytoplasm will be shed off, and the remaining part of Golgi plus remaining cytoplasm are shaded off and the phagocytosized by the Sertoli cell. This is an important function of the Sertoli cell. And when the spermatid try to develop into the spermatozoa, its excess cytoplasm are phagocytosed by Sertoli cell, and then the spermatozoa are released. And this is basic structure of spermatozoa. You can see the elongated nucleus, and just above the nucleus the acrosome cap are present. Also the spiral mitochondria are developed, and the flagella are also developed, and this flagella later extended from the middle piece up to the end of tail. Then this is the process of spermatogenesis, and by which million of sperm are synthesized in our testis or in our reproductive organ or the primary sex organ. And in next video we will discuss about structure of ovum. So thank you, and that's it.